In this module and next several modules, we look at the process of cell division. We will look at the process and components, machinery that allows cell to make a copy of itself. As we know, all organisms have a limited lifespan. So in order for life to continue, cells will have to make more copies of themselves. As we have already established, cell is the basic unit of life. Cell division, the process ensures the continuity of life. From one cell, we can make more than one cells or two cells. Unicellular organisms propagate by splitting into two cells. We look at that process also. Multicellular organisms, organisms that are made up of many cells, they need cell division to replace old dead cells or senescent cells in order to keep their organs and tissues functional and viable. In humans, billions of cells die each day. For example, red blood cells, our epithelial cells, these cells have to be replaced. The replacement of the cell has to come from another cell in the process of cell division. Additionally, a special type of cell division called meiosis, we'll talk about this process, this kind of cell division in more detail later on. This ensures the organisms shuffle its genes, allowing different combinations and allowing natural selection to take place and selecting organisms with best combination of genes. Let's start this with the simplest organism. Let's look at the process of cell division in the simplest organism, a bacteria, a prokaryote. We have already mentioned that these organisms don't have any membrane enclosed organelles, which means they don't have a nucleus. They have a nucleoid region, which we talked about, which contains DNA. DNA in these organisms is attached to special positions in the membrane, the cell membrane. Bacterial cells can divide as long they have favorable environmental conditions. So as long you provide bacteria with food, they will keep on dividing. Let's look at this process. The membrane is part of the membrane, special part of the membrane attaches to the bacterial DNA. That's where a special sequence of DNA is. This special sequence of DNA is the origin sequence. That's where the DNA replication starts. As we have already mentioned, that genetic information is contained in this type of polymer, the DNA. So when a bacteria has to divide, conditions are favorable. First of all, the DNA will start replicating. It will start making a copy of itself at the ori point of origin of replication. And as the cell grows, it stretches. Now initially we had only one ori, now we have two oris. As the cell elongates, the two oris will also separate and segregate. Once the DNA replication is complete, a cell wall membrane will form and which will separate the two cells. So initially we had one cell, from one cell we have gone to two cells in this process. We look at this process in multicellular organisms in the later module.